to welcome Pall Thordason, Fellow of this Society and leader in the field of complex chemistry, to deliver his presentation. The slide shows Pall's formal credentials, which are significant and extensive. But in the spirit of adding more colour, I can tell you that Pall was raised on a sheep and cattle farm in the northeast of Iceland and always intended to continue the family tradition as a farmer. But in a first for his family, he decided to go to university and graduated from the University of Iceland with a BSc in 1996. He told me he's not quite sure why his path took him to Australia for the first time to do a PhD, but thankfully for us, he did. His PhD supervisor was Max Crosley, who is well known to the Society as an important contributor and continues to be so, but he's now also contributing to the French wine industry. He told me recently, when I managed to email him by mistake instead of Merlin Crosley, that in February this year he made 62,000 bottles of great wine at his vineyard in Rochheim in France during the August 2023 vintage, but is still writing papers of importance. Pall's PhD, which was awarded in 2001, centred on the self-replicating properties of porphyrins. Armed with a Marie Curie fellowship, Pall completed a postdoc in the Netherlands, which initiated his interest in RNA. Taking his next role in Australia in 2003 at the University of Sydney was a conscious decision of Pall to return to our shores. And the rest is history, albeit still in the making. Currently, Pall is director of the UNSW Sydney RNA Institute, program leader for the New South Wales RNA Production and Research Network, and leader of the New South Wales RNA Bioscience Alliance on behalf of the New South Wales UNSW Vice-Chancellors Committee, or the New South Wales Vice-Chancellors Committee. He's also president of the Royal Australian Chemical Institute. Tonight, Pall will tell us why RNA is having its place in the sun after a long period, almost 1,322 meetings, of fundamental research. We're aware of the importance of RNA because of the efficacy of the RNA vaccines in the SARS-CoV-2 pandemic, and of course the Nobel Prize awarded to Jennifer Doudna and Emmanuel Charpentier for their development of the CRISPR-Cas9 system, a revolutionary gene editing tool that allows us to rewrite DNA. Some of you, as did I, may have attended Jennifer Doudna's presentation a couple of weeks ago in the sold-out concert hall of the Opera House. It'd be great if the Royal Society of New South Wales could mount such an event. There's a challenge for you. So please welcome Pall tonight for his presentation, RNA and Me, from the origins of life and nanomedicine to building an RNA ecosystem. Pall, welcome.
Thank you, Susan. I'm very deeply honored to get the opportunity to talk to you here tonight. I should say I'm a good personal friend and of one of my probably key mentors who was a president of the society some years ago, Bryn Hibbert. Unfortunately, he couldn't make it here tonight, but I feel like I would not be here tonight if it wasn't for Bryn. And Bryn was the person who actually introduced me to this society, and I joined it. I can't actually now quite remember, about 5 to 10 years ago, so I'm sure Bryn could look it up. And one of the deepest honors I ever had was when I was made a fellow of the Royal Society of New South Wales, so I just want to thank you again for that. Right, so I'm going to try to take you through a bit of a journey of RNA. I'll sort of divide this talk into probably 3 components. First one will focus in on my own personal research journey in this space. Which is, like, I guess, a lot of my other life and work, very non-linear. You can see how you probably wouldn't have predicted when I was 20-year-old on a farm in Iceland I would end up here. But anyway, such is life. And maybe with sort of reflecting on it, that's why I'm interested in complexity. I don't seem to do things in a simple way. So I'll move on from my research and sort of take a bit of a journey into, of all things, science and politics, and then try to go on from there to what we actually want to try to do to improve society and advance science with both the RNA Institute and also both the state and national-based initiatives in RNA. Right, so back to basics. In terms of why suddenly the world has, as you said, RNA has come out of the darkness. I argue it's because RNA is the key tool for gene medicine, which is a fairly diverse range of emerging technologies. But I guess one question you might want to ask yourself, why is RNA so important then, particularly for this new field of gene medicine? Well, I'm going to borrow from Moderna's CEO the phrase that RNA is a software of life. Now, all analogies and metaphors are dangerous, right? But what he said is that you could, and sort of, I'm going to change slightly what he said. You could imagine if the DNA is the hard drive where the key information is stored. We know if we have a computer device, the hard drive doesn't do much on its own, right? The storage. What makes your computer do something useful is the software. And RNA is the software. The machine reads the hard drive and creates software from that, which is the RNA. And something which is very important here too, and I could have been, converted by John Maddock, those of you know him. RNA is not just the messenger. Yes, mRNA, the messenger RNA, is the most famous part of RNA, but actually, just like software, mRNA is the software that sends a message to the printer to print out a protein. But we have a lot of all the software generate in the cell from the DNA. And I think for me personally, both as a chemist and also as a scientist, that's actually where the excitement is, not about mRNA. And from a chemist point of view, as I say, it codes for a lot of all the things than just proteins. And there's all these other forms of RNA. Some have been known for a long time, the tRNA, ribosomes, and so on. But there's all this other non-coding RNA in it. John was actually one of the first people to point out they're not junk. They have a function. The interesting thing is we're still trying to understand what that function is. And from a health point of view, we're now starting to realize more and more. A lot of diseases, particularly complicated diseases, complex diseases like neurodegeneration, many genetic influence diseases and whatnot, are probably at the root cause. There's some misregulations then in these RNAs, right? From a chemist point of view, they're also fascinating molecules because structurally they're much more diverse than DNA. DNA is kind of boring. It's either double strand or single strand or nothing else, right? RNA is a much more dynamic molecule. Now, the structural biologists hate it also from that point of view because it's really impossible now to work out the structure. But yes, it has a lot of diversity. And my good friend Matt Disney at the Scripps Institute in Florida, he's made a living as a medicinal chemist on trying to design molecules that can control the interactions of all these different forms of structural RNA with proteins. Something that when he started at 10, 15 years ago, people said he's nuts. But of course, he proved everyone wrong and has a couple of startup companies already in that space treating diseases. Now, so as I said, the first part of my journey is kind of about our research right now, mostly, but with a bit of a prehistory too. So before I forget, I just want to acknowledge that people actually do the real work. I don't really do anything useful. And that's my research group. And also kind of what motivates me sort of from time to time to continue is to get the opportunity to work with these wonderful people, as well as the different organizations that have supported my work throughout the last 15, 20 years. So in our research group, a key component of our work is the RNA peptide droplets and gels. And this, you'll see this a bit more details. We kind of also came into this to some degree, work that started about 2010 with the Australian Centre for Nanomedicine, the UNSW, that Marie Cavalaris and Justin Gooding and Tom Davis set up, and I was a part of from day one. And working with Marie and others on creating better delivery systems, including for RNA. So And then we're also doing some really cool work in light switchable gels and cellular engineering, but I'm probably not going to talk much about that now. So just to sort of give you a flavor of what we were doing with Maria and Josh, and was to create ligands that target particular cell types associated with medulloblastoma cancer, right? So this was a piece of our work back in the pre-COVID times, making this peptide conjugating to a cancer drug. Now what happens then is that doxorubicin, which is still used in cancer therapy, is one of these horribly cytotoxic cancer drugs. It's very active, but it's also very non-discriminate, and that's why it's cytotoxic. When we linked our peptide to it, we shut down the uptake of that drug to normal cells, but we maintain it to cancer cells, because now we get targeting towards cancer cells. So that was sort of where we're heading. And then we had these polymer nanoparticles that we decorated with those same peptides. And we could move on then to show that if we used our control cell lines, if we put peptides on our particles, we get a bit better uptake. But with the target cell, we get a huge discrimination. So that's the idea of making delivery systems that target particular cell type. Now, this is still the main challenge in any RNA therapeutics. You go to industry and ask them, what's the main challenge with RNA therapeutics, be it vaccines or cancer therapeutics based on RNA? Delivery, delivery, delivery. It's about getting your drug molecule in the form of RNA of some kind into the right cell type and or tissue type, and obviously also at the right time. That is not trivial, and we're still a long way from solving all those challenges, but it's fun too. And now, so in the last 2-3 years after establishing the institute, we moved more on to the LNP, lipid nanoparticle platform. This is the same platform that the mRNA vaccine that many of you know are made by. It's actually a really simple process. You mix a solution of your nucleic acid, mRNA or other RNAs, with ethanolic solution of the lipids, and in a process of, which is sometimes referred to as nanoprecipitation, you get the nanoparticle and encapsulate the RNA. So what we're doing now is to taking conventional LNPs, like in the vaccines, and trying to link our peptide to the surface to increase the cellular specificity. And I literally, at 3 p.m., I couldn't add it to the talk, I saw some fantastic results from our collaborators in Joshua McCorrell's lab using our nanoparticles, where they say, wow, okay, they're actually working. So watch this space, yes? But this keeps me excited when I'm dealing with bureaucrats and whatnot, right? So I said origin of life. So this goes back to, and I said to you, Susan, I have absolutely no idea still, and my mother was very shocked when I said, I'm going to Australia. I still don't know why, but anyway. It did have to do with Max Crossley, because my boss in Iceland knew him from his Oxford days and said, hey, I thought you said something about you interested in going to Australia. Yeah, I know this nice guy in Sydney, his name is Max Crossley. And as they say, the rest is history. Now, Max put me on this project on self-replicating porphyrin. That's all about really origin of life. We want to understand how molecules can make copies of themselves. Now, like all the good PhD projects, that part of it failed, but some other part worked, right? But I got the origin of life bug from that. I was fascinated by this field from day one, and I was on the slide trying to do research in this space. In fact, the nanomedicine work I was doing was kind of trying to do origin of life by stealth, by making protocells, because I knew I would never get funded for it. So now, as a chemist, this bit is but a lot of people get excited about how did the first biomolecules like nucleobases emerge from that pre-biotic soup. Actually, I'm more interested in the next step. Once you go to slightly more polymeric, like peptides and RNA, how do we end up with something like this? This is, to me, the key molecule we need to understand. How did we get a proto-ribosome where we had simple-ish peptide and simple-ish RNA interacting together, where you get simultaneously information transfer and catalysis happening? There must have been a simpler version of this somewhere on this journey, right? So I always wanted to go back to this ever since my PhD. Now, liquid-liquid phase separation, the simplest phenomena in somewhat in physical chemistry. one that you're familiar with when you mix vinegar and water, or your lava lamp. There's a slightly more interesting version of this with two solutions of the same solvent mix, and obviously water being the most important. Now this image is not from Iceland, it's from Alaska. So what's happening here is you've got the ocean and you've got a glacier river. So you've got two aqueous systems with very different ionic strength, because there's also polymer clay in here. What happens then is that they don't mix, at least not immediately. So you get phase separation between two aqueous phases. And it's a lot more common than people think it is, in part though because these things are driven by kinetics. They're not thermodynamically stable systems, but as you can see here in this case, they can last for miles, i.e. hours. So now when people start looking at the cell, obviously we see the membrane-bound mitochondria and the nucleus. There's all these other blobs here. And as microscopy techniques improved, people start to look more harder at these blobs. And then, thanks to Paul Kermey, in 2016, I came across Archie Fox's work. Now, she is from Sydney originally and then went to UWA, but she discovered these so-called paraspeckles in the nucleus in the early 2000s. They are one of these so-called membraneless organelles, and they are actually liquid-liquid phase-separate systems within the cell. They're made out of proteins and RNA, a non-coding RNA. And paraspeckles now are one of the best characterized of these systems. So Archie and I started sort of collaborating in 2017. This is really what dragged me back into the RNA world. And so this system sits in a nucleus. They're about 500 to 1,000 nanometer in size. They consist of two particular non-coding RNA called NEAT11 and NEAT12, and a bunch of proteins. And this is not a stoichiometric system. This is an aggregate. This is like a nanoparticle inside the nucleus. And there's a bunch of these things, and they control cell, gene expression and whatnot. Again, we still don't fully understand some of that. And it's interesting, too, that the proteins involved in these and other droplets have a lot of these sort of unfolded, intrinsically disordered region with so-called prion-like sequences. So, from a polymer chemist point of view, these are coacervates. It's negatively charged RNA complexing with a positively charged protein. That's an oversimplification, but it's a starting point to understand these systems. Why does it matter? Well, one of the reasons is why it seems to matter, and there's more and more evidence coming from that, is that these droplets in the cell, sometimes when they go wrong, we might be getting aggregates. So perhaps, this is still not settled, this is the ultimate cause of many neurological and other disease indications where we see protein aggregates. Because often the question is, what are these proteins doing there in the first place? They are building blocks for these droplets, and those droplets in turn are organizational elements within the cell. This is a quiet but very interesting revolution that's coming through biology in the last decade or two, and I say, watch this space. So we come from the chemistry point of view, and we're also very fortunate in 2018 to hire Albert Fahrenbach as an academic staff member at UNSW. He came from the Solstack lab at Harvard and brought RNA synthesis, something I had never done, into our school, and we started collaborating. And those who are into chemistry, it's similar but different from peptide synthesis. I love it, but it's sort of, if that's your cup of tea, I could go on and on and on, but probably shouldn't. The point is you can make large-ish, up to 80 long RNAs. Now, we started with something very simple, though. Grace was my first student on this project. We made short RNAs, pentamers, with one nuclear base in the middle, varied, so like mutants of AAAA and AACAA. And we made this really short peptide based on these sort of protein structures we saw, really simple systems. And we just mixed them together. And guess what happened? We got droplets. So even very, very simple RNAs and peptides form these droplets under the right conditions. And you can immediately see here, just visually, the only difference here, same peptide, same concentration, one nuclear base. There's some differences. And we then moved on to sort of base our work around particular family of peptides mainly, the so-called arginine, glycine motifs, which are very common, these intrinsically disordered proteins. And in the work that Grace did then as a follow-up, for instance, she can see, now we're going to quantify this aggregation here on the y-axis. We can see that as we increase the concentration of the peptide, we can see different behavior depending on the length of the RNA, whether it's a decamer or a polymer, which is about 50 to 100, we get different aggregation. Same with the uracil-based ones, so these multimers of RNA. Annika then took this to a slightly different direction, and she made 2 different RNAs. One is a decamer of AG repeats, so the purines. One is a decamer of the CE repeats, pyrimidine. There's just a difference in the nucleobases in terms of aromaticity, nothing in the charge. We mix it with a charged peptide, you would not expect any difference. Well, obviously, and you can actually now see fluorescently labeled there, so we can see them grow, really cool. But there's a difference. These guys form aggregates, these don't. So it's not just about charge, yes? So this is the stuff that keeps me excited as a supramolecular chemist, because we're really trying to decode these interactions at the submolecular level. So, right. And Adiba has taken a slightly different view where we use a molecule we know we can switch with light between 2 conformers. Now, first of all, before she did that, she also looked at 2 regioisomers. Hey, organic chemistry 101, regioisomers, orthomethopara, hey. So you would not think they make much of a difference for this particular peptide, but they do. We get quite different aggregation behavior just based on the 2 regioisomers. Then she took the para one, and she starts with the E form, so this is like trans cis, right? And we can switch between them with light. So with UV light, we go from the trans to the cis, and with green light, we can go back. So we can see actually that they have different aggregation behavior, and probably more dramatic this way. So we take the droplets, we shine UV light, they disappear, and then we shine green light, and they come back. And we can go back and forth like this 10 times or more. So we can cycle it this way. One example of our fundamental work is this work that Lachlan Koss is doing, and is a collaborator with Felix Risotto, a very clever young man working in DNA origami. And we're now interested in sort of moving beyond the classical Watson-Franklin-Crick pairing to Hookstein-based pairing, because this is a bit of an oversimplification of how RNA folds. There are other interactions than just those normal base pairing involved, particularly the so-called Hookstein pairing. This involves, say, potassium ions between 2 guanidines, or at lower pH, 2 cytidines can also complement each other. Right. So the best known examples of these are so-called G quadruplexes, which both DNA and RNA can form in the presence of, say, potassium. So we made this RNA and it forms a quadruplex. We're also looking at these I motifs, which is a lesser known relative of these guys. Very similar, but slightly different 3-dimensional structure. What's really interesting then is when Lachlan takes this charged peptide here, is that it binds very strongly to the folded I motif for RNA and the folded G motif, but not to the unfolded. So just the folding of the RNA determines whether it binds to a particular peptide. So that's just sort of where we are right now today. So I'll start off here with sort of the work we're doing right now. There's a lot of questions. How important is sequence selectivity, the mixing order? This is all kinetic trapping or something else. For pre-biotic chemistry, there's a lot of questions about salt concentrations, pH, and so on, chirality, secondary structures. So this is what keeps me motivated, because it's fun, right? Science has to be fun. So I say to people, come and join the RNA peptide world. There's so much fun in there. Now, that's going to be my cue to switch to the second part of my talk. I might just grab a little sip of water. This is a rendering of the Pfizer mRNA molecule. It's a very large molecule. Do you realize it has a molecular weight of about 1 million? So if you have a protein, Peter, an mRNA is always 10 times bigger, roughly. Like, you don't think about it, but so your GFP is 33 kilodalton, the mRNA for this is 330. Fascinating when you start to think about it that way, right? Now, outside the success story of mRNA vaccines, this didn't just happen overnight, because RNA therapeutics has been actually a field growing and growing over decades. And particularly small interfering siRNAs. There's been billions of dollars invested. Oops, sorry, that wasn't what I was going to do. There's been billions of dollars invested in that. There are all types of therapeutics here, both in terms of forms of RNA and also in terms of indications and where these things are in terms of development, registration, and so on. But of course, the vaccines in 21 was just like another dimension. The profits for these two companies in that 1 year were eye-watering. Now, I should say, not so good for them right now. Like, not that they are poor, but the glory days of those profits are over for the time being. So it's why the industry is sort of now moving on from, okay, great, we solved that problem, now we need to do something else. And that's actually a good thing, because it means they're going to push on to cancer therapeutics and other things. For me as a scientist, I also got thankfully familiar with this curve long time ago, actually through solar cell work we were doing on the site. The technology readiness level idea. It came from NASA. Basically, here's your idea, here's your basic research, here's your pilot scale, here's your production, sort of pre-production scale, and here's your product. In medicine, roughly speaking, this is the valley of death. This is the later preclinical scale, which is tedious and very expensive and hard to get funding. Academia is hard enough, but this is harder. Once you get to human trials, although they get much more expensive, it's easier to raise funds. What Australia has missed out on for a long time in this space, and Peter could probably tell you about this, is not only we have been underinvesting here in the preclinical, but we don't have a lot of manufacturing in this space, so it all happens offshore. So Queensland actually in the protein space led the way with the Pantheon facility to try to reverse that, but too late in terms of Ian Fraser's work, of course. So of course in 2020 in RNA space, well, there's just nothing here, right? But there's a bit of a history here, and RNA, again, in Australia, is actually something we've been doing very well for a long time, and I love this example from the 80s, the work the CSIRO was doing on ribosomes. There's some patenting on this, the hammerhead ribosomes back in 1987. And there's a bit of a connection here to this room, with this, because we have to follow me here for a second. So Gene shares a CSIRO spin-off, and there's an article here in the AS Eureka, plus Susan gave me this sort of filled in some of the information. Hopefully this is all roughly correct. Gene shares was established in 87 in partnership with this French company, Johnson & Johnson Research, where you worked, Susan, for a long time, then joined in soon after. And for nearly 2 decades, led the development of this technology until JJR closed down here. That's not the end of the story, actually, because there were 2 spin-offs that kind of came out of this. SpeedX, which I think many of you know, Alison Todd's company, which is very successful, and Calmion, which Jeff Simmons set up, and then CSL bought him out, and now gene technologies is one of the 3 main pillars in CSL. So this story is now creating billions of dollars worth of economic activity in Australia. So we didn't just start yesterday, right? But that being said, pre-COVID, there wasn't really a lot going on, at least in 2019. We had CSL doing a little bit. They actually also owned some very interesting IP in this space. Didn't do much with it. And then Sue Fletcher and others in Perth have been working a long time on antisense oligo technologies. And out of that came a company called PYC Antisense in Perth, which is still doing very well. But that was about it. And of course, in terms of R&D, well, that was it, right? Not a lot, right? So I had met Archie, as I told you, back in 2017, and it was kind of thanks to her. She grouped me into this spontaneously self-assembled group in July 2020. And we kind of just started trying to help this gentleman, Colin, developing an mRNA vaccine in Monash, but ended up accidentally being a lobbying group for RNA. And there's something, I'm not hesitant to say this anymore, but in that period, mid-2020, we came to closed doors everywhere. With one of the exceptions was the New South Wales Chief Scientist's Office. But the message in Canberra, we have now found out later on, basically, the powers to be in Canberra in mid-2020 had convinced themselves that mRNA was an unproven experimental technology. And it wasn't until very late that year they changed that. So this is, I think, why not only did we miss the opportunity to say help him doing it much earlier and whatnot, but possibly also why we ended up at the back of the queue for vaccines compared to Europe. But anyway, that history should be written one day. And we expanded this group to include our friends at UQ, Trent Munro, and then when he left, Tim Mercer. And it's been a fantastic avenue to keep communication going in what I'm sure you noticed has been a bit of a state-to-state fight. But individually, we also tried to keep some sense to that. And we got this right up in 21. We won the hearts, minds, and dollars. I'm not sure that's a compliment, but it was interesting. So the backstory here, as I say, is that we started a nanomedicine center. I mentioned it briefly, and there was a center of excellence here, very strong, we were involved in Albert. But in 2019, for me, was a critical moment on that journey. Unfortunately, Chris Armstrong is not here tonight. I've known him for a long time, and thanks to him, I met him and then Hugh Durham-White back in 2019. And essentially, Peter actually said, we lost duration proteins to Queensland and Victoria. I think RNA is next, we should do that. And they said, well, that's an interesting idea, but that's all that happened. But then 2020 came along, and I started getting phone calls from them. So, yes, it sort of snowballed from there. And then I should have added Gabriel Upton was very key person to get the government interested. And then, yes, once the Premier got interested, then the proverbial shit hit the fan. And what was unusual about that initiative was that Premier Berejiklian insisted on the universities working together on this. And that's why the New South Wales ACT Vice Chancellor's Committee was the partner. Barney Glover did absolutely heroic work on that front. And then he put me in the hot seat to work with Hugh to actually deliver this. Politics and some changes, and I ended up there at the press conference when we announced the factory. Here's Gabriel and Ian Jacobs, VC at UNSW. Yeah, and morning sun with photochromic glasses, I look like a bodyguard, so I'm not going to do that one again, I think. I should point out there's been a bipartisan support for this here in the state, and we're exceptionally happy to see how the new government here is also continue supporting this. And what we came off basically is this scheme here. This is my drawing of it. There's an investment in a factory which is going to be built by the state government and operated by Myeloid at Macquarie Uni, hopefully breaking ground in the next few weeks. And then a 10-year R&D investment by the state with the universities. It's quite unusual, and plus all this collaboration. And then each university has decided in some cases to do its own things, like ANU and us. New South Wales Health helped us putting up this network also in 21 between 5 of the universities, sort of kicking things off. And as I said, that's about the factory. We'll hopefully get a bit more, I know there will be some public announcements about this fairly soon, because it's all ready to break the ground now in the next few weeks, I hope. I think this is the more interesting one, because this is Hugh Durham-White's idea of how we can get the university and state government to work together. It's essentially a bit model on a CRC, but different, where the state government and the universities have put their investment into some long-term R&D programs. So this is called RNA Australia, and again, you will see a bit more about that soon. Now, at UNSW, our Vice Chancellor Ian Jacobs supported me in setting up an institute. It builds on the old Nanomedicine Center, our analytical center, the Kirby Institute, which works on HIV, Ramaciotti, and the CCI. And quite unusually, we brought together people from 3 faculties. And scientifically, we're trying to address challenges in biology, medicine, and chemistry. And then our facility, our core facility, we call it the accelerator, which provides access to preclinical materials of all different kinds of RNA. And just to show you who is who involved in the institute, there's 30 academics. Some of you might recognize some of these faces, but I'm very proud of having the opportunity to work with this very diverse group of chemists, biologists, chemical engineers, biomedical engineers, cancer experts, infectious disease experts, medical scientists, and a pediatrician, just to name a few, and a psychologist as well. UNSW has particular strength in delivery, so that's kind of one of the key things we're trying to do, but we're also into manufacturing from a quality control and analytical chemistry point of view in particular, sensing as well, and so more fundamental interactions of RNA with proteins, peptides, and so on. And we also, as part of this initiative, appointed 3 new people in these different fields here. I'm just trying to rush through this a bit. So, are we going time? Okay, not too bad. This maybe also gives you a bit of a taste for what RNA can be used for. at UNSW, just to give an example. And I will say across the country, there's so much else going on. So this is just sort of a snapshot from one university. So we're looking at using not mRNA, but silencing RNA as an antiviral, as a next generation antiviral, particularly for immunocompromised people. Those of you not familiar with the Kirby Institute, it started off as the Australian HIV Institute, so they've been working on HIV for 30 years. We're also working with people at the Garvan Institute at second generation vaccines. Maria and co at the Children's Cancer Institute have been working for probably close to 15 years now on interfering RNA as a potential tool for treatment of cancer in children. Plus, there's a good program there too in the medicine on pancreatic cancer. Michelle Farrer is a pediatrician who treats kids with these diseases like spinal muscular atrophy with RNA therapeutics as well as viral vectors. And there's now a Sydney-based rare disease center emerging between Randwick and Westmead in this space. And that sort of links to this, and Jennifer Doudna's visit actually. Westmead is building a GMP factory in the viral vector space, but we're also recognizing that for Sydney, combining that with the RNA factory, we can do both modalities of CRISPR, either the viral vector one or what's called a non-viral based one, which is using mRNA and guide RNA. And so we have a lot of interest in that space. And people like John Marek and I, at least we think the future is actually going to be neurological conditions, and not just neurodegenerative, but possibly also things like schizophrenia and even addiction, as well as autoimmune diseases. And there's a lot of interest too in linking genomics, AI, and development together. So the facility, so we can make the plasmid and that are required then to make the mRNA, and we can also, and this is kind of our unique angle, we do also oligonucleotide chemical synthesis, and then these can be combined can be put into lipid nanoparticles. This accelerator facility was the first RNA facility in Australia to get an ISO 9000 certification. We have now 18 staff members. We've done over 300 products. And as I say, work across this space. My favorite example as a chemist is that uniquely we have mass spec capabilities in the space. And I think mass spec is where it was in proteomics 20 years ago, and it's going to be complementary to all the sequencing methods to understand better the complex structure of RNA in biology. Because conventional sequence methods, even the Oxford nanopore method, don't pick up all the chemical modifications that our bodies do on RNA, and there's a lot of them. So where are we? It's amazing. It's just actually amazing where we are now in Australia. I'm not sure this map is complete, but this is my attempt to map all the companies that I'm aware of, big and small, doing something in the RNA space. I include New Zealand for a reason. We have a very good relations with them in this space as well, and in some ways we're one economical zone. I'll pull out from that sort of the bigger pieces. So the four big pharmaceutical companies, because they're kind of in a different category because they're kind of mostly doing their own stuff, and that's obviously very important. But for startups or academics, if we want to work with them, we probably end up most of the time handing over IP. Now that's works for some, but not for others. So Moderna, of course, is the federal government investment. It is expensive. We are talking several $1 billion. But it's a national security asset because it means hopefully the next time we have a pandemic, we can make it locally. And even if it's several $1 billion, that's only a fraction of one nuclear submarine and probably much more useful. So Now, CSL is really coming into this hard now because they actually had a lot of very important IP in this space anyway. The Victorian government has also invested in bringing BioNTech in with a bit of a smaller operation, but it's going to be important. And then Sanofi in Queensland has a partnership with UQ and the Queensland government in Griffith, which I'm very excited to see where it will go. But again, it's centered around what Sanofi wants to do. The red ones are different because they are kind of CDMO type operations that are agnostic to where people will come from. And they are at various stages. So Biosina is an old site that's been bought up by another company and is converting to RNA. ITT does formulations in Melbourne. South Pacific Sera is also a GMP factory in New Zealand that is pivoting in that direction. And then here in Sydney, we are setting up this myeloid operation with state government. So what's this space? Now, on the research side, we have the base facility at UQ, We have our institute. We have RNA Australia, which brings together everything here. The Shine Dalgano Center, ANU, fantastic research center in RNA biology. We have Innovation Hub collaboration with Monash in Melbourne, plus the mRNA core at Monash, Colin Powton. And Arthur Fox and friends have established this center here over in Perth as well. Plus, there's the RNA technology platform in New Zealand. Last thing, just to some of the last things we have done recently. I'm very proud of this. We made Australia's first mRNA animal vaccine. It's an interesting partnership. It's led out of Menangle. Really, Jim Rothwell should take, probably is the person who should be credited with doing most of this, and is funded by New South Wales Department of Primary Industries. The IP and development comes from a small company in Boston called pre-biotic, spin-off from Bob Langer's lab on dendrimer technology. So different from Star Pharma, but yep, it is what it is. They includes the Canadian Inspection Agency, because we see in a moment, because of the animal expertise we need. And yes, what it's really for though, kind of brought back on here, this is the real reason we're doing this. We want to be ready with an emergency response if there's an outbreak of foot and mouth or lumpy skin disease in Australia, because none of the other GMP factories are ready for another 1 or 2 years. So we've been working on this at UNSW. We've been working with APMA. We're working, of course, with TIPA. We're initially using this sheep disease here, with this Menangle people have a lot of experience in, as a sort of a crash test dummy. And it's working. I mean, and then we made our first local batch a couple of months ago, handed over to Menangle, and we showed that it's actually, if anything, better than the one TIPA was making. So, because our job is to upscale the TIPA process, and that is working. So we are now in a position, if needed, throwing resources, we could upscale that emergency production probably within weeks, if necessary. I'm hoping we don't have to, but it's a real biosecurity threat. And as an ex-farmer, this is also something that's very close to my heart, right? So. Now, Team Australia, like as I say, to the outside world, it always looks like this is just one of these bloody state competitions, but it's not quite like that, thankfully. And Therapeutic Innovation Australia is a really good example. And Peter, you know this one very well, of course. So what we've done here is to bring together some of these centers under one hat. We have just launched 2 weeks ago this TIA increased RNA protease capability, so linking together in collaboration what's happening at UQ here, Monash, and UWA, to help everyone to go further in this field. So I just want to thank all my collaborators at UNSW, my research groups and funders, and you for your kind attention. Do we have time for questions? Or? Yes. Yes.
So firstly, I really like hearing Jim Rothwell's name, because I work at DPI New South Wales.
There, I'll take the question.
Sorry.
There should be can somebody run a microphone around? Thank you.
Yeah, I was just going to say it's great to hear Jim Rothwell's name, because I know him from my work, which is DPI New South Wales. So that was really cool. I'm quite interested. I read a paper, I think it was by Ira Devison or Martin Smith in the last few years, about RNA as a sort of organelle, which is a really different sort of paradigm to what's been presented in classical biological training, right? And I just wonder, with the advent of AI technology, whether you can see a future where we can design predictive RNA therapies that will fundamentally change the RNA state in cells and really do drastic changes to biology and in positive outcomes for humanity.
Yeah, that's a really interesting point. Because sort of from my own research, the reason I'm interested in droplets is that that's exactly what I think we are starting to think more and more. They're almost like organelles. They don't have a membrane, but they're organizational units within the cell. Now, from that, you can see John Mardiger argued this. This is maybe how cells communicate, and cellular development in embryo and whatnot is actually regulated, is through these. You can then go on from there to therapeutics. And that's kind of also why we're looking at this peptide interaction. We can understand how to do it. Now, AI will come into this. I don't think it's a magic bullet. I do a lot of nonlinear regression in my research, and AI is just linear regression on steroids, and that's why it hallucinates, right? So as long as you got good training data, the problem in RNA, and we just had a company called RNA Fold AI moved to UNSW from New Zealand. Some really bright people run that. Problem is the training data we have now is really poor, which is why AI is not quite there, but they're trying to push the envelope. But actually what we need to do is to do more structural chemistry and biology to generate the training data so we can then use AI to develop what we need to do. So it will play a role, but you can't do it in isolation from collecting more training data. And compared to proteins, we're much poorer when it comes to RNA.
Questions? Peter Gray.
Well, thanks, Pali, for a really excellent presentation. And I think where you ended up, if you look around the world, Australia is very widely and positively regarded on two schemes, the Collaborative Research Scheme, CRCs, and the National Research Infrastructure, NCRIS, National Collaborative Research Infrastructure. And you ended up talking about what you're doing in the RNA field, where you and three of the groups around the country have got together what's almost a model for national research infrastructure, where you're sharing standard operating procedures, quality systems. So you're really working as an integrated national research infrastructure system to help researchers translate their research to new therapeutics. So congratulations, well done.
Thank you.
A great talk. Thank you.
Thank you.
Questions? Can you tell us about Jennifer Doudna's companies and Jennifer Doudna's companies and the attacks, the lines of commercialization they're following?
Yeah, so those of you who came to her lecture, I mean, she does have she was actually explaining to us, she's still doing fundamental work and then they got applied work. What was very interesting is that for CRISPR, again, the main challenge is not the CRISPR RNA technology per se, it's the delivery. So if you want to target a particular tissue or whatnot to do gene editing, you need to get the CRISPR system one way or the other to that end. So that's the focus of her lab. Here's another Icelandic connection. There's a guy called Daniel Oskar Frímannsson, and he works now for her company called Mammoth Biosciences, and that's exactly what he's doing there, trying to help them developing better delivery systems, because that's now the bottleneck in CRISPR, and I would say in gene medicine. And I'm not shy to say this, when we were doing I was in a learning exercise with the government to work with the government and do a business case, as an academic. What the heck is that? Oh my God, that was fun. But in the business case, we had to do a business case, of course. On one hand, we made the argument, and I think successfully, that Sydney's unique angle compared to the rest of Australia and the world is probably in gene medicine. And obviously there's a lot of clever things happening here in vaccines too, but I think that's where the real cutting edge is for us, plus delivery system. And that goes to work in nanomedicine, not just at UNSW, but Wollongong, Sydney Uni, and ANSTO. ANSTO actually has some very good unique capabilities in that space. And that's why we want to try to capitalize on in that sort of international competitive environment, because again, even in the gene editing field, the limiting factor, the rate limiting factor is delivery.
Anyone else? Because I've got another question. We've just had $1 billion investment in PsiQuantum from the Australian government. When's it going to come for RNA?
Okay, what can I say?
I should disclose.
Yeah,
I'm on the board of the Sydney Quantum Academy.
This is in the public domain. I was one of the people that was put. I was very honoured. I was put on the external advisory group that Kathy Foley put together last year, with Archer. Actually, Archer was one of them. John Skerritt and others. Yeah, and there was this was run by DICER, Department of Industry, Science and Resources. So that's Ed Husik's department, and it was to kind of start a national conversation about, what next with RNA, particularly where could the federal government come in on the top of what the state governments have done and the initial Modern investment. All I can say is we did a lot of work, and there's a lot of paperwork that is now with DICER, and I'm crossing my finger. Maybe we'll get some very good news from Ed one day. Maybe not. I mean, I've been in this now long enough to realize that you just have to wait and see. So.
Very diplomatic. Any other question, Guy? And then up the back.
Thank you very much for your presentation. You've talked around the messenger RNA and the CERN and that aspect. What about in the smaller, the 40 to 800 nucleotide and the spacing and information? Because traditionally a lot of the research and science has been on the genes, not so much the non-coding. Your fluid was very, very interesting, but are we starting to look at just how important that space or lack of spacing may be?
That is music to my ears as a chemist. If you're looking for a good scientific sort of science book now to read in the public domain, I would encourage you to read a book called How Life Works by Philip Ball. He's a former editor of Nature. He's sort of like me, converted now. I think biology needs to move on from the sort of digital view of biology. Genes are not the answer to everything. There's higher level of order within the cells between the macromolecules, RNA, proteins, and so on. And that, from a chemist's point of view, it's a fuzzy one. And the three-dimensional and time, what's really four-dimensional, three-dimensional and time dimension of how these molecules interact is actually what matters in terms of how the cell develops and survives and interacts with other cells. And it's an emergent property of these molecules that sits above the DNA level. And I think the biologists, I think some of them have, like Archer and others, but some not. We need to move on from this idea that we can solve all the problems in biology just by hacking gene data. That is not possible. I know it's going to be important and we'll do a lot with it, but that's only the first layer. There's another layer of hierarchy above that we need to start to work much harder on. So I love that comment.
Thank you. Is there a microphone for... If you could tell us who you are.
My name's Alan, first time at a Royal Society meeting. I was from one in 2018, but then COVID shut all the meetings down. So. You mentioned that the RNA technology was being used in the addiction space. You mentioned that RNA technology may be used in the addiction space.
Yes.
Just wondering, are you targeting like substance addictions, like opioid or alcohol or nicotine, or will it also function for process addictions like gambling? And
now, I have to admit I don't know too much about this, except Associate Professor Kelly Clements at Psychology at UNSW. She works on short interfering RNAs. She can inject rats with it that have been made addicted to cocaine, and they stop being addicted. Now, that is pretty much all I know. I don't fully understand how that works, but that's just to give you a taste of. So there are people like her and Tim Brady at UQ and others, and again, John Maddock is one of the prophets of this area, say like, to understand the brain, we need to actually start to look at RNA as well, much harder. That's maybe where a lot of the organizations and for instance, maybe that's how long-term memory to some degree is encoded, is in these sort of structures that RNA forms. So I'm not entirely sure what Kelly is doing. I'm not entirely sure she would know all the details, but she's worked out particular interfering RNA injected into an addicted rat. Doesn't like having cocaine. Go figure.
Thank you.
Don.
Just following on, I'm Donald Hector. Just following on from Susan's question about quantum computing and your answer to the question from Guy there about the emerging systems nature of these systems. What's the potential for quantum computing to model these systems and to provide much more responsive answers to the questions?
Now we hit something very interesting. So AI on its own, yes, I think it would be useful. I just happened to have the conversation with Michelle Simmons about this just literally weeks ago. As a chemist, we also be waiting and trying to help Michelle and others, like, can we use quantum technologies in general to also understand chemical systems, because they're quantum-based? And we all know that that hasn't quite still happened. We should be able to, and this is what Michelle's been working on for over a decade and others. I think we're almost there. But by extension, if we can do that, we can also start to look at the structures of RNA by the same sort of quantum algorithm. And actually, I think it's probably more likely there will be a step change in our understanding of RNA in biology through quantum computing than AI. That's just my gut feeling. But that's not going to happen until the quantum people have cracked that nut of doing proper quantum mechanical calculations on chemical systems that are bigger than a few atoms with their quantum computing. And yeah, she will be the first one to admit we're not quite there. We're almost there, I think. Jagadish probably would know more about that than I do, but it's almost there, isn't it? Yeah.
One last question. No? Good. So please thank Pali for a wonderful presentation.